So welcome, everybody. I apologize for the slightly delayed start. We had some technical issues, but I hope that this is all sorted out. So welcome. I'm Jörg Ehrman. I'm a rheumatologist at Brigham and Women's Hospital in Boston, and I'm currently the vice chair of Spartan. This is the second seminar in a series of 6 global education seminars organized by GRAPPA and Spartan. And I'm proud to mention that during the first seminar a month ago, participants from 32 countries actually participated. The recordings are also available on the GRAPPA and Spartan websites for more people to watch. For today's seminar, we've had registrations from more than 40 countries, and I would like to welcome you all. So good evening. We have evening here in Boston. Good morning or good afternoon, wherever you are in the world. Today's seminar will focus on the genetics and pathophysiology of psoriasis, psoriatic arthritis, and axial spondyloarthritis. Because of time limits, we had to make some selections with regard to content. So for instance, we decided not to discuss the role of intestinal microbiota. So please let us know in your post-seminar evaluation what you think about our selections and whether the volume and also the degree of detail were appropriate for this program. Next slide. Next slide. This shows us the agenda for today's program. So Niti Goel will start off the seminar. After that, we'll have 3 presentations from Kurt de Vlaam, Louise Puig, and Chris Richland. Each of these presentations will be framed by a couple of multiple choice questions. We are going to collect the data anonymously, so please participate, even if you're not 100% sure what the correct answer is. But your responses will help us who develop the educational content for this program in future sessions. At the end of the seminar, we'll have a live question and answer discussion, and all the presenters will be available for this session. And so please stick around. And I also would like to encourage you to type your questions to the presenters into the chat window. Next slide. So the last slide of introduction, I would like to thank Pfizer for financially supporting this program. Next slide. So the first presentation will be by Niti Goel. Dr. Goel is a rheumatologist at Duke University in North Carolina. She is also a patient with psoriatic arthritis, and she is going to talk about a patient perspective on genetics and pathophysiology.
Hi, I'm Niti Goel. I'm a patient research partner from the United States, and I'm also a rheumatologist. I first began experiencing psoriatic arthritis in December 2009. Although I diagnosed myself with psoriatic arthritis within 6 months after the first onset of acute symptoms, it was at least 3 years before I sought treatment for it formally from another physician. In general, with my psoriatic arthritis, I do not have significant psoriasis. I have had a dark patch on my left elbow, as well as what I call defective fifth toenails ever since I was a child, but these were not recognized as being potentially part of my psoriatic disease until I developed the arthritis. My arthritis symptoms started with a cold that I experienced, and I woke up one morning with lower back pain. Shortly thereafter, I had a massage scheduled with the massage therapist who noted that the area on my back, which hurt, was warm to touch. Then over the next month, I developed bilateral heel pain, then my hands and wrists swelled up, and I had pain on the balls of my feet. I initially thought I had axial spondyloarthropathy, but subsequently I started to get nail changes in both of my big toenails, which was onycholysis. That is what tipped me off that I had psoriatic arthritis. I didn't seek help right away, mainly because I needed to get some personal things sorted out in my life, but I kept symptoms at bay by self-treating with medications that didn't work very well. Subsequently, after I finally got my life issues sorted, I did go to a rheumatologist. Once I started treatment that actually worked for my psoriatic arthritis, I realized that a lot of symptoms that I had had since I was a teenager, like locking of my left elbow, pain in my jaw, and neck pain, were probably related to the psoriatic arthritis since they resolved with the treatment. Though I know of no one else in my family with psoriatic arthritis or axial spondyloarthropathy, my children have had some early symptoms of disease. However, it is too soon to diagnose them with anything definite, though one has had dactylitis and jaw pain. I have, however, had physicians give me information related to my children in terms of their genetics that doesn't necessarily apply to them because I am of Asian Indian ethnicity. I would like healthcare providers to be better about discussing the genetic diversity of this condition and to relay information with the appropriate context related to the genetics. I would also like to know my children's risk of disease and if there's anything I could do to prevent the condition worsening or developing at all in them. In addition, in speaking with other patient research partners, things that were important for us to understand related to the pathogenesis and genetics of these diseases were the triggers for flare and increased disease activity, prognosis, how to influence disease outcomes, and predictors of treatment response, as well as the possibility of individualized therapy. When discussing data with us, please make sure to be clear about recognizing if we're a member of a population at risk. If the study included, for example, mostly individuals of European ancestry, acknowledge the findings may not be applicable to those of us of different races and ethnicities, and clarify any assumptions that you've made based on the available data or lack thereof. Thank you so much for allowing me to share my story and perspectives as a patient research partner.
Next slide. So we'll have now two pre-questions before the next presentation, and this is question number 1. An HLA-B27 positive woman with ankylosing spondylitis comes to the clinic accompanied by her 6-month-old son. Assuming that the son is also HLA-B27, what is his approximate risk for developing ankylosing spondylitis? Only one of the answers is correct. A, 0.2 to 1%, B, 2%, C, 20%, D, 50%, or E, 90% if the father is also HLA-B27 positive. You have 5 seconds to answer. 5, 4, 3, 2, 1. Next slide. Pre-question number 2. Which of the following statements about the psoriasis risk allele HLA-CO2-602 is correct? A, it is absent in indigenous populations in South America, Southern Africa, and Australia. B, it is a major risk factor for all types of psoriasis. C, it is associated with a lower risk for HIV and HCV infection. D, it confers an increased risk for the development of psoriatic arthritis. Or E, it is associated with a better response to treatment with ustekinumab. Again, 5 seconds to answer. 5, 4, 3, 2, 1. So here the answers are more spread, and we'll post the questions again at the end of the presentation. So the next presentation is by Kurt de Vlaam. Dr. de Vlaam is a professor of rheumatology in Leuven in Belgium. and he will discuss the genetics of psoriasis, psoriatic arthritis, and ankylosing spondylitis, or axial spondyloarthritis.
Welcome to the session about genetics in axial spondyloarthritis, psoriasis, and psoriatic arthritis. We will discuss the role of genetics in the susceptibility of the different diseases in the different clinical phenotypes and other potential applications. I have no disclosures to this presentation. The spondyloarthropathy concept groups a number of different clinical entities with common clinical, immunological, and genetic features. Clinically, you can divide the group into predominantly axial spondyloarthritis and predominantly peripheral spondyloarthritis. Ankylosing spondylitis is the hallmark of the axial group. Psoriatic arthritis is typically representing the peripheral group. Clinical features include spondylitis, sacroiliitis, peripheral arthritis, enthesitis, and dactylitis. Patients can also present extra-articular manifestations such as colitis, psoriasis, and uveitis. Besides ankylosing spondylitis and psoriatic arthritis, IBD-related arthritis, juvenile spondyloarthritis, reactive arthritis, and undifferentiated spondyloarthritis belongs to the concept. They share also genetic features, topic of this presentation, and they have inflammatory pathways in common, resulting in overlapping treating modalities. In the early 70s, HLA studies indicated an increased association between some HLA genes and ankylosing spondylitis and psoriasis. The association of ankylosing spondylitis with HLA-B27 was found almost simultaneously by two different groups, just before HLA-W17 was found to be associated with psoriasis. Studies for ankylosing spondylitis about disease concordance in twins and families, and also later in psoriasis, confirmed that genetic factors largely contribute to the susceptibility of these diseases. This is nicely illustrated by the fourth generation pedigree you see from the Iceland family affected by psoriasis. In the case of ankylosing spondylitis, the hereditability is estimated around 90% in the twin studies and confirmed around 63% in later GWAS studies. For psoriasis and psoriatic arthritis, the hereditability is somewhat lower, but all these findings underscore the importance of the contribution of different genes in these diseases. There is a group of diseases that seems to be associated with a number of MHC class I molecules, the so-called MHC class I-opathies. As in the case of AS, we see HLA-B27. In the case of psoriasis, we found HLA-CW6. In the case of ulcerative colitis, we found HLA-BW4. But also other diseases like Behçet disease are associated with an MHC class I molecule. It is clear that in this group the strongest association is the one between ankylosing spondylitis and HLA-B27. Over 85% of AS patients are positive for HLA-B27. This also highlights the fact that some patients with AS are not positive for HLA-B27. In that case, an association can be found with HLA-B60, now called B40. Recently, also an association with one of the familial Mediterranean fever genes is reported in a Turkish population. Interestingly, the association with MHC class I molecule suggests also some mechanistic clues. MHC class I molecules, such as HLA-B27, are expressed by all cell types and play a role in antigen presentation. The MHC class I pathies have some common features and are completely different from other autoimmune diseases like rheumatoid arthritis and systemic lupus erythematosus. These autoimmune diseases, they show a female predominance, there is no liquid injury and there is never gut involvement, and they are associated with MHC class II molecules. For joint disease, autoimmune disease have most of the time synovial involvement and a polyarticular distribution, while the diseases belonging to the MHC1 nopathies have most of the time involvement at the sites of mechanical stress. This means at the antithesis and in the lower limbs. Some years ago, McGonigal suggested a new classification with a continuum between monogenic autoinflammatory diseases at the one end and monogenic autoimmune diseases at the other end. In between, you find polygenic autoimmune and autoinflammatory diseases, and some diseases show overlap with autoinflammation and autoimmunity, such as ankylosing spondylitis and psoriatic arthritis. The actual hypothesis is that gene variants can cause immune dysregulation after mechanical stress or interaction with environmental factors, which are ubiquitous, such as infection, trauma, and repetitive loading. So, what kind of genes are we looking for? Well, we look for genes that increase susceptibility, genes that are associated with certain clinical features of phenotype, such as articular disease, extra-articular disease, or even comorbidities, and genes for disease outcome or treatment response. Genetic variance modeling has indicated that genetic factors contribute to over 90% to the overall susceptibility in ankylosing spondylitis. HLA-B27 shows one of the strongest associations of an HLA molecule with the disease and is the most important gene for developing ankylosing spondylitis. Another very important gene seems to be ERAP1, but variations in ERAP1 only contribute to the disease development in the presence of HLA-B27, a phenomenon called epistaxis. Thus, ERAP1 alone do not increase the risk for developing ankylosing spondylitis. ERAP1 as a molecule trims peptide to the optimal length before being loading on the MHC class I molecule. The association with both HLA-27 and ERAP1 shows that the antigen presentation and processing might play an important role in the pathogenesis of spondyloarthritis. Since these both genes do not confer the complete genetic risk, other genes must be involved. Other genes actually definitely associated are IL-23 receptor, a family member of the kinesin family 21B, an IL-1 receptor, anthraxin receptor 2, and STAT3. For other additional genes, confirmations in different populations is still needed. You should be aware that most of the actual results of GWAS studies were performed in patients with an European or Chinese ancestry and are not by definition generalizable to all AS patients from any ethnic origin. So additional studies in other populations are needed. The involvement of IL-23 receptor, kinesin family member 21B, IL-1 receptor, anthraxin receptor 2, and STAT3 implicate that genes related to innate immunity and to the Th17 pathway also play a role in the susceptibility of ankylosing spondylitis. The variation in IL-1 receptor 1 and 2 alleles points towards the innate immune response, and the allele variation in IL-23 receptor and STAT3 show that regulation of the Th17 pathway plays also an important role in the susceptibility. Also, genes in the acquired immune response like RUNX3 play a role in the CD8 T cell function are important. HLA-B27 is not only associated with ankylosing spondylitis but also with the other spondyloarthropathy members, but to a lesser extent. The highest frequency is found in juvenile spondyloarthropathy and undifferentiated spondyloarthritis, and to a lesser extent to the axial forms of IBD-related arthritis, psoriatic arthritis, and reactive arthritis. The association of HLA-B27 with AS is found in almost all ethnic groups around the globe, but the prevalence is varying amongst the different ethnicities. The lowest prevalence and thus the lowest association is found in the sub-Saharan region. This does not mean that AS doesn't exist there or is not present, but it is present to a lower extent and possibly associated with other HLA types. Recently, an association with HLA-B7 and B40 was reported in this region, but still need confirmation. Actually, over 100 protein coding HLA-B27 variants exist, but not all are associated with ankylosing spondylitis. HLA-B27, 05, 02 and 04 are definitely associated. HLA-B27, 06 and 09 are definitely not. The others, it is still unknown and need further research. The risk for developing ankylosing spondylitis is related to the presence of absence of B27 and is best studied in the Caucasian population. The frequency of HLA-B27 in normal Caucasian individuals ranges from 8 to 40%. HLA-B27 in Caucasian AS patients is present in over 90% of the patients. The prevalence of AS in this population ranges from 0.2 to 1.4%, and the prevalence of ankylosing spondylitis in random B27 Caucasian individuals is 2%. But the prevalence is much higher in B27 positive relatives of AS patients and is almost absent in B27 negative relatives of AS patients. So, already mentioned, absence of HLA-B27 do not exclude the diagnosis of AS, but it is clear that HLA-B27 positive patients differ in many aspects from HLA-B27 negative patients. HLA-B27 positive patients has an earlier age of onset and earlier age at diagnosis. They have a greater familiar occurrence. They are more associated with acute anterior uveitis, but less with psoriasis and IBD. They show a better response to TNF inhibitors and show an association with ERAP1. For psoriasis, we see that in the 80s, HLA-CW6 has been recognized as the PSOR1 gene and confers to the susceptibility of psoriasis development. Nonetheless, CW6 is not associated with all aspects of the skin disease. CW6 is associated with psoriasis, but not with scalp psoriasis, nail psoriasis, or intergluteal psoriasis, the major risk factors for developing psoriatic arthritis. CW6 is part of a haplotype associated with early onset type of psoriasis, type 1 psoriasis. A haplotype is a combination of genes on a unique chromosome which are transmitted together. Additional alleles in this haplotype still must be defined. Again, the association of an HLA class 1 molecule points towards a role for antigen presentation in the susceptibility of psoriasis. Since the identification of CW6, many more genes were found associated with psoriasis and psoriatic arthritis. Actually, more genes associated with psoriasis are found than with psoriatic arthritis. The challenge to find genes associated with psoriatic arthritis is that actually most genes found in psoriatic arthritis are also associated with psoriasis and thus not unique for psoriatic arthritis. Actual findings, mainly based on genome-wide association studies, show that different biological pathways are implicated in the susceptibility to psoriasis and psoriatic arthritis and its pathogenesis. Besides genes playing a role in antigen presentation, the regulation of Th17 pathways and innate immune response, genes implicated in the skin barrier function like LCE3 and corneodesmin, and genes in the interferon type 1 signaling pathway contribute to the susceptibility. At the moment, few genes are exclusively associated with psoriatic arthritis. There are associations with HLA B38, 39 and B27 and also genes in the IL-1 gene complex and with variants of IL-13. One of the problems in PSA is the phenotypic heterogeneity and the case ascertainment. Actually, we have some HLA association with the different clinical phenotypes in psoriatic arthritis. Joint deformity and ankylosis are associated with HLA-B8. Dactylitis is associated with the haplotype B8B27, and enthesitis is associated with B8 also, and all this compared to patients with cutaneous psoriasis or controls. So, why should we go for identifying more genes? Well, genetics has proven to be a powerful tool to improve genetic risk prediction, to find new drug targets, and to identify novel pathways that provide important clues to the pathogenesis and newer treatments. To date, most genetics are studied in respect to the disease susceptibility, but genetics must go beyond susceptibility. Various strategies can be employed to study genetic factors that influence the complete conceptual trajectory from risk of psoriasis through disease onset, prognosis to patient outcomes, and other aspects you see as onset of disease, severity, comorbid burden, and response to treatment. Recently, data became available of a potential genetic association with treatment response. Patients with the CW0602 allele have a better and longer response to ustekinumab compared to the patients who were negative for CW0602. Within this C0602 group, patients having additionally the CO4 genotype has significantly more chance to have a sustained PASI 100 response compared to the other groups. Actually, we are living a genetic revolution with the development of new technologies and new analytical methods leading to more global projects, including studying different populations. In the recent years, the combination of GWAS with meta-analysis has led to the discovery of many new genes, especially genes with a lower allele frequency. I like to conclude stating that genetics in spondyloarthritis, psoriatic arthritis, and psoriasis are rapidly evolving due to the advent of new technologies and methodologies. It is unclear if genetics will improve the diagnostics, but recent findings help to better understand pathophysiology, and recent findings helps also to develop new drugs and to make a more optimal treatment choice for our patients. Thank you.
Thank you very much. Before we have a very brief Q&A with the speaker, let's do our post questions. So let's go back to question number 1. Next slide. So this was the question about the genetic risk in the 6-month-old son who is HLA-B27 positive of a mother who is also HLA-B27 positive. So here are the answers. I'm not going to read them. You have 5 seconds to answer. 5, 4, 3, 2, 1. Okay, excellent. So the third option is, of course, the correct answer. And I think that question may have been a little too easy. But it seems that HLA-B27 is not such an easy thing, and we may get back to this in the Q&A session. Next slide. So which of the following statements is correct about the psoriasis risk allele HLA-CO602? A, it's absent in indigenous populations in South America, Southern Africa, and Australia. B, it's a major risk factor for all types of psoriasis. C, it's associated with a low risk for HIV and HCV infection. D, it confers an increased risk for the development of psoriatic arthritis. Or E, it is associated with a better response to treatment with ustekinumab. And you have 5 seconds. 5, 4, 3, 2, 1. And the correct answer here is E. If we can go to the Next slide. So this is what we heard about in the presentation, and maybe we can also discuss that. Welcome. Maybe I can actually ask you that question first. So what are your thoughts about the mechanism? How does the HLA haplotype predetermine the response to therapy with an anti-cytokine antibody?
Well, the class 1 molecules plays a role in the antigen presentation. So up to now, it's by let's say, although the problem is that we don't know the specific antigen which binds to these class 1 molecules yet. But what we know is that, in fact, binding of the antigen can elicit a inflammatory, in fact, starts an inflammatory cascade in this cell and may lead to the inflammatory process. So if a specific molecule, monoclonal antibody, is blocking this process, this can partly explain the association with specific HLA molecules.
Yeah, so MHC class 1 plays an important role in the spondyloarthritis and in psoriasis, but we don't understand the mechanism. So the HLA-B27 association was described in 1973, so almost 50 years ago. Why is it so difficult to figure out what the MHC does?
Well, the problem is with the HLA-B27 molecule is that it's in a zone, in the region where there are many other genes which might play a potential role in the disease process. Although we think that HLA-B27 might play directly a role, we are not 100% sure. Maybe it's one of the genes next to it, which segregates together with the HLA-B27, which might play a role. There are different hypotheses about HLA-B27 or the role of HLA-B27 up to now, but still it's unclear, and many people are still working on it. But it's not completely, let's say, understood yet and proven what the role is. Another argument is that, as mentioned, there are now more than over 100 different HLA-B27 proteins, and only a couple of them are associated with the disease. And we think that this especially has to do with typically antigen binding, but upon binding, it's still unclear what the exact mechanism of this association is. Yeah.
Okay, I think we have to move on, and we'll have more time at the end of the seminar for questions. Let's go to the next pre-questions. Next slide. Next slide. So this is pre-question number 3. According to current pathogenetic models, what does the initiation of psoriasis have in common with physiological responses to skin injury? A, strong activation of IL-36 and IL-17C production by keratinocytes. B, innate response mediated by mast cells and polymorphonuclear cells. C, type 1 interferon production by plasmacytodendritic cells. D, the activation of myeloid dendritic cells via TNF. Or E, the upregulation of autoantigens such as ADAM-TSL5 and LL37. And you have 5 seconds to answer this. 5, 4, 3, 2, 1. Great, I was hoping for that. So this will be very educational. Let's go to pre-question number 4. Which of the following antibody target pairings is incorrect? A, bimekizumab and IL-17RA receptor. B, brodalumab and IL-17C. C, spesolimab and IL-36 receptor. D, risankizumab and IL-23p19. Or E, ustekinumab and IL-12p40. And again, 5 seconds to answer. 5, 4, 3, 2, 1. Okay, thank you. And with that, I'm handing over the microphone to Dr. Luis Puig. He's going to give us all the answers to these questions. Dr. Puig is a professor of dermatology in Barcelona in Spain, and he is going to talk about cytokines, disease phenotypes, and drug targets.
Hello, my name is Luis Puig. I'm a member of the scientific committee of GRAPPA, and I'm going to talk about cytokines, disease phenotypes, and drug targets. There are no disclosures for this presentation except the sponsor of this whole educational program. And the topics that will be covered are the cytokine networks in psoriasis, focused on psoriasis vulgaris, the phenotypic and endotypic correlation and therapeutic interventions, with some comment on psoriatic arthritis and spondyloarthropathies. Whenever dermatologists look at typical plaque psoriasis, the histopathological correlation pops up in their minds. Keratinocyte proliferation determines typical psoriasiform hyperplasia of the epidermis. Their altered differentiation determines thickening of the stratum corneum with retention of nuclei. And their immunologic activation releases chemokines and lymphokines that attract polymorphonuclears and lymphomononuclear cells and determine dermal edema, blood vessel dilation and proliferation, and signs of ongoing inflammation. The initiation and maintenance of psoriasis can be considered an unrestrained activation of physiological responses to skin injury, such as the type 1 interferon-dependent response to microbiota and skin injury, which activate toll-like receptors and determine a robust innate immune response with increased expression of antimicrobial peptides, which also occurs in psoriasis. On the other hand, the adaptive arm of immunity is also activated by autoantigens such as ADAM-TSL5 or cathelicidin, also known as LL37, and even interleukin-26, which bind DNA and would activate toll-like receptors in plasmacytoid dendritic cells and lead to interferon release and activation of myeloid dendritic cells, which would present those antigens both in the lymph nodes and in situ to naive T cells. This early phase of psoriasis vulgaris would be predominant in acute and stable or erythrodermic variants, rather endotypes, of psoriasis. In post-streptococcal guttate psoriasis, there would be a prominent adaptive response by a restricted repertoire of T cells directed against streptococcal epitopes shared by keratinocytes. On the other hand, stable lesions of plaque psoriasis would be defined by a marked adaptive response with predominant activation and persistent polarization of T17 lymphocytes driven by interleukin-23, which is produced mostly by myeloid dendritic cells. T17 cells release interleukin-17A and interleukin-17F, which synergize with tumor necrosis factor alpha to activate the immunologic response of the keratinocyte, whereas interleukin-22 would drive the epidermal proliferation. Activated keratinocytes would release chemokines and lymphokines that attract several types of immune cells, amplifying the inflammatory response. Mast cells and polymorphonuclear leukocytes would contribute further preforming interleukin-17 and release proteases that would activate pro-interleukin-36 produced by keratinocytes in a self-amplified loop. Interleukin-1 and interleukin-36 would be especially relevant as regards expression and maintenance of the pustular endotype characterized by predominantly polymorphonuclear intraepidermal collections. Whereas interleukin-12 and tumor necrosis factor have amplifying roles, interleukin-23 is the main regulatory cytokine in psoriasis and drives differentiation and maintenance of T17 and T22 populations. Interestingly, tissue-resident cells expressing interleukin-23 receptors are responsible for fast relapses of psoriasis in previously involved but apparently cured locations by producing high levels of interleukin-22 and interleukin-17. Interleukin-22 determines the keratinocyte proliferative response in association with interleukin-26 and interleukin-29. Interleukin-17A and F also activate the keratinocyte to amplify the inflammatory response and are also responsible for endothelial changes and other manifestations of inflammation in psoriasis. The role of tumor necrosis factor is synergistic with interleukin-17, partly by increasing the expression of interleukin-17 receptors in the membranes of target cells. Immune-activated keratinocytes release numerous chemokines and lymphokines that attract both innate and adaptive immune cells and potentiate the inflammatory infiltrate. Keratinocyte production of interleukin-36 and interleukin-17C create a self-amplifying loop that promotes an increased expression of antimicrobial peptides and the release of a wide spectrum of pro-inflammatory cytokines that contribute to epidermal proliferation as well as to local and systemic inflammation. Thus, in the last decade, there has been a shift in focus in our understanding of the pathogenesis of psoriasis from T-cell activation driven by antigen presentation with IL-23 as the main regulatory cytokine, towards a focus on innate immunity with a central role of the keratinocyte not only as a target, but also as a producer of cytokines that activate dendritic cells, promote local T17 polarization, and participate in self-amplifying loops. Furthermore, the role of polymorphonucleus and mast cells contributing large amounts of preformed IL-17 and proteases that activate pro-interleukin-36 is highly important. The role of IL-36 as a pathogenic mediator of inflammation and epidermal proliferation in psoriasis was initially recognized in the imicimod-murine model, but found its final confirmation in the identification of monogenic variants of autosomal recessive psoriasis or sporadic cases of psoriasis with some phenotypic variation, but characterized in most cases by the appearance of widespread pustules and systemic symptoms corresponding to autoinflammatory diseases. This disease is due to loss-of-function mutations in the interleukin-36 antagonist gene that determine unrestrained activation of the interleukin-36 receptor. Only 1/3 of sporadic cases harbor these mutations, but those who do have earlier onset and more systemic symptoms. Generalized pustular psoriasis has been known for many years with three main phenotypes: early onset, to which most of the monogenic cases correspond, late onset or von Thunbusch disease, and impetigo herpetiformis in pregnant patients. A recent proposal of classification highlights the systemic component of the disease, its frequent association with psoriasis vulgaris, and the relapsing versus persistent course of the disease. Localized variants also exist, either with palmoplantar or involvement or destruction of the nail apparatus, and can coexist with the generalized form in one given patient or in different members of the same family. The histopathological hallmark of the disease are intraepidermal pustules with variable dermal inflammatory infiltrate and epidermal hyperplasia, depending on the evolution of the lesions. Several additional mutations have been recognized in pustular psoriasis variants, and there is some phenotypical variability in members of the same family and even in a given individual. But the net result of these mutations is enhanced activation of nuclear factor kappa B in the keratinocyte, which determines release of interleukin-36. Keratinocytes are the major source of interleukin-36 in the skin, and the expression of interleukin-36 can be induced by other pro-inflammatory cytokines, including interleukin-1, tumor necrosis factor, and interleukin-17A. Interleukin-36 is secreted as full-length pro-interleukin-36 that becomes cleaved by neutrophil-derived proteases into a truncated form that has more than 500-fold greater biological activity than the pro-interleukin-36. This truncated interleukin-36 can act back on keratinocytes via the interleukin-36 receptor to induce even more interleukin-36 expression, as well as expression of neutrophil chemokines. The latest identified mutation inactivates an inhibitor of neutrophil proteases. This cartoon summarizes the central role of the keratinocyte in driving localized and systemic inflammation. Interleukin-17, interleukin-1, and tumor necrosis factor alpha induce release of chemokines that amplify this loop, as well as self-potentiating loops of interleukin-17C, interleukin-17E, tumor necrosis factor alpha, and the cytokines of the interleukin-20 family. Interleukin-36 becomes activated by neutrophil proteases and skin-derived cathepsin and signals on keratinocytes, monocytes, and other cells, leading to release of interleukin-6 and systemic inflammation. Interleukin-36, combined with other keratinocyte-derived cytokines, activates dendritic cells to express interleukin-36 that acts on plasmacytoid dendritic cells, inducing release of interferon alpha and further systemic inflammation. Dendritic cells release interleukin-1 beta, interleukin-6, and interleukin-23, which close this adaptive immune cell loop. The recognition of the central role of interleukin-36 in pustular psoriasis and polymorphonuclear infiltration in psoriasis has put the focus on the cytokines of the interleukin-1 family with their complex activating and inhibitory roles, which seem to be active both in psoriasis and in psoriatic arthritis, as recently reviewed by the Pizzalis group. Therapeutic drugging of this pathway has started to provide very promising results in generalized pustular psoriasis with at least one monoclonal antibody directed against the interleukin-36 receptor, spesolimab, in phase 3 of clinical development. As a matter of fact, the application of translational research to the therapeutic armamentarium in psoriasis has yielded extraordinary results. The first class of molecules were directed against tumor necrosis factor alpha, but their efficacy in psoriasis was at least matched by ustekinumab, directed against the P40 subunit of interleukin-12 and interleukin-23, with an excellent safety record and a very advantageous frequency of administration. The advent of interleukin-17 antagonist pushed PASI 90 response rate beyond 70% at 12 weeks. The efficacy of these drugs in psoriatic arthritis and spondyloarthritis is comparable to that of adalimumab. Secukinumab and ixekizumab must be administered every 4 weeks and require high induction in the first year. Brodalumab blocks the receptor IL-17RA and thus signaling by multiple isoforms of IL-17. Bimekizumab, a dual inhibitor of interleukin-17A and interleukin-17F, is in advanced stage of clinical development. Another dual inhibitor nanobody, sonelokimab, is also under development. The latest class of a specific P19 or interleukin-23 inhibitors is generally pushing the complete clearance rate beyond 50% to 60% at 16 weeks. They are administered every 8 to 12 weeks with good maintenance of response over time and persistence of the therapeutic effect beyond pharmacokinetic expectations, that is, remission, potential disease modification, and a safety record that matches that of ustekinumab. This network meta-analysis illustrates the short-term and long-term efficacy of the available drugs for the treatment of psoriasis. Of note, the very low placebo response rate at PASI 90 or PASI 100 levels of clearance. The best performers, ixekizumab and brodalumab among the IL-17 inhibitors, and guselkumab and risankizumab among the P19 inhibitors. Risankizumab seems to lead the pack over long term, but the clinical relevance of this evidence in real life remains to be seen. Thus, the interleukin-23, interleukin-17 pathway has proven to be of central therapeutic importance in psoriasis. Interleukin-17 antagonists have the fastest onset of action, especially brodalumab, which inhibits the interleukin-17 receptor A and the signaling of multiple interleukin-17 isoforms, followed by ixekizumab. Guselkumab and risankizumab are P19 inhibitors characterized by high levels of clearance, which is maintained over long term and decreases slowly upon discontinuation. The median decay of PASI 90 response is 24 and 36 weeks since the last dose, respectively. From the safety perspective, TNF-alpha blockade may decrease antitumor immunity and depletes granulomatous response against intercellular pathogens such as Mycobacterium tuberculosis. IL-17 inhibition is associated with increased candida infections and some risk of inflammatory bowel disease exacerbations. whereas interleukin-23 inhibition is devoid of these concerns, probably because of redundancy of the immune response. Interestingly enough, newly developed oral agents blocking the relative subunit of tyrosine kinase 2 and thus intracellular signaling by IL-23 and other cytokines might provide an alternative option with similar efficacy to ustekinumab and an acceptable safety profile in clinical trials. This table summarizes the differences between classes of biological agents targeting cytokines in psoriatic disease. Of note, drug immunogenicity decreases drug survival in anti-TNF-alpha agents and contributes to the fact that other classes be the preferred choice by dermatologists. Psoriatic arthritis and spondyloarthritis share with psoriasis many pathogenic cytokines, but the target cells are mesenchymal stem cells and osteoclast precursors, and the final activity inducing bone erosion and/or bone formation depends on modulation by interleukin-1, interleukin-6, and tumor necrosis factor, as well as rank and its ligand. On the other hand, mechanical trauma is the main trigger of the inflammatory response in the enthesis and the chondro-osseous interface. Many cell types producing T17 cytokines are potentially independent of regulation by interleukin-23, which would explain the differences in response to interleukin-23 and interleukin-17 inhibitors, especially on axial disease. I would like to wrap up with some take-home messages. Both innate and adaptive immune responses are involved in the pathogenesis of psoriatic disease and spondyloarthritis. Interleukin-17 is the main effector cytokine contributing to both inflammation and structural changes. It acts in synergy with tumor necrosis factor and is mainly regulated by interleukin-23. Activated keratinocytes in psoriasis release multiple chemokines and cytokines that amplify inflammation and epidermal hyperplasia. Interleukin-36 in particular is quintessential in driving pustular psoriasis. Translational research has led to the development of several classes of biologicals with unprecedented efficacy in psoriasis. Cytokine networks in psoriatic arthritis and spondyloarthritis are similar to those involved in the inflammation in psoriasis, but anatomical differences and specific cell types involved in bone remodulation contribute to differences in the efficacy of available treatments. Finally, I want to finish with a view of my hospital in Barcelona City, which I hope you can visit soon. Thank you very much.
Thank you very much. That looks indeed like a great visiting target once the pandemic is over. So let's go to our post questions. So according to current pathogenetic models, what does the initiation of psoriasis have in common with physiological responses to skin injury? A strong activation of interleukin-36 and IL-17C production by keratinocytes. B, an innate immune response mediated by mast cells and polymorphonuclears. C, type 1 interferon production by plasmacytoid dendritic cells. D, activation of myeloid dendritic cells via TNF. Or E, upregulation of autoantigens such as ADAM-TSL5 and LL37. 5 seconds to answer. 5, 4, 3, 2, 1. So still some uncertainty about the correct answer, and we'll welcome Dr. Pusch in a second to clarify this. So the correct answer here is C, type 1 production by plasmacytoid dendritic cells.
Yes, it is the innate immunity activation through toll-like receptors of plasmacytoid dendritic cells that drives an increase in the expression also of antimicrobial peptides. And type 1 interferon is a driver not just of the response to injuries, but also to the acute and early and more, let's say, unstable variants of psoriasis. So early on, type 1 interferon response is what drives psoriasis, as well as paradoxical psoriasis and some other unstable variants of the disease.
Yeah, I think the critical point here is really what is similar between early psoriasis and a response to injury, because some of the other answers are correct with regard to other phases of psoriasis.
Yes, exactly.
Okay, next slide, Q4. So which of the following antibody target pairings is incorrect? A, bimekizumab and IL-17RA, B, brodalumab and IL-17C, C, spesolimab and IL-36 receptor, D, risankizumab and IL-23p19, or E, ustekinumab and IL-12p40. 5, 4, 3, 2, 1. Okay, I think we have a significant change compared to the pretest. And next slide. And this is the correct answer. Can I welcome Dr. Puig here on screen? Yes. Hello.
Hi. Hello.
With regard to this last question, I think that there is a need to, and your presentation helped with that, to really sort out what the different IL-17s are. Can you, because you mentioned IL-17C in your presentation, can you sort of clarify what the relationship is between IL-17C and A, and whether the currently available antibodies, in particular secukinumab and ixekizumab, block that target?
The question wanted to stress the point that bimekizumab blocks both. It's a dual inhibitor of both IL-17A and F, so it has nothing to do with the receptor. On the other hand, brodalumab has a wider blockade, not just of IL-17A, AF, and F, but also of interleukin-17C, which is recognized currently as a very important driver of psoriasis because it's produced by the keratinocyte itself. And it also is able to block other cytokines of the IL-17 family or isoforms, which might or might not have an pro-inflammatory or anti-inflammatory role in both type 1 and type 2 responses. But the important thing is that by blocking IL-17C, this could explain the faster action of brodalumab when we compare that to, for instance, secukinumab.
Okay. There was a question earlier in the chat, and I don't know whether everyone followed that, and that was about interleukin-22 as a therapeutic target. Yeah. Can you comment on that?
Yes, the secukinumab was developed by, I would say it was Pfizer early on. Not Pfizer, it was Wyeth, I would say. And it was tried, and it did not work in psoriasis. The main reason for that is that IL-22 is important in proliferation, but it's overcome or driven by interleukin-17. So by blocking IL-22 alone, there are many redundant pathways by which keratinocytes can proliferate. IL-17, IL-19, there are many other more important cytokines in that respect. And the same applies to atopic dermatitis, as a matter of fact, because the cytokines responsible for the acanthosis of the epidermis in the lichenified or chronified forms of the disease are also interleukin-22 and interleukin-17. And interleukin-22 blockade works only in those patients with the highest levels of interleukin-22.
Okay. So, one last quick question, if there's a quick answer. What is the mechanism of the paradoxical induction of psoriasis that we see in particular patients treated with TNF inhibitors?
It seems to be quite complex, but in the bottom line, there is an increase of interferon alpha production that again switches back this chronic adaptive response in psoriasis towards a more unstable disease with increased production or release of interleukin-17C and interleukin-36. So eventually, when there's a paradoxical psoriasis, we need to block either interleukin-23, which is activated by interferon, or interleukin-17, and even perhaps in the future, interleukin-36, which is so important in the mechanism of production of pustules in paradoxical psoriasis. You all know, based on your experience in rheumatology and also in inflammatory bowel disease, that these patients are severely handicapped by the pustules that develop on their palms, on plants, and also sometimes on hairy in the follicular areas.
Okay, thank you very much.
Thank you.
Let's move on to our last presentation for today. But first, a couple of questions. So question number 5, next slide. Which of the following statements about tissue-resident memory T cells is correct? A, these are long-lived cells that continuously circulate through non-lymphoid tissues. B, these cells have a limited T cell receptor repertoire. C, these cells are lymphoid cells, but they do not have a T cell receptor. D, they express CD69 and CD103. Or E, they provide a very rapid antigen non-specific cytokine response to local injury or infection. 5 seconds to answer. 5, 4, 3, 2, 1. Okay. And question number 6, next slide. Which statement describes best the role of IL-22 in psoriatic arthritis? A, it provides a key proliferative signal to dermal fibroblasts in the skin. B, it is a major mediator of pathologic new bone formation in the spine. C, it is a critical activator of osteoclasts in peripheral joints. D, it mediates the recruitment of regulatory T cells to sites of inflammation. Or E, it may play a limited role outside of the skin. And you have 5 seconds to answer. 5, 4, 3, 2, 1. Okay. And with that, I invite Dr. Chris Richland. He is a professor of rheumatology at the University of Rochester in New York, and he will talk about cell-cell interactions in the pathogenesis of psoriatic disease and spondyloarthritis.
Hello, I'm Christopher Richland, and I'd like to talk to you about navigating the immune landscape in psoriasis and spondyloarthritis. I have no disclosures for this presentation. I'd like to talk about psoriasis and SPA pathogenesis, focusing on interactions of distinct cell populations through activation of multiple pathways. I'd like to discuss these interactions and pathways, emphasizing tissue complexity and cellular plasticity, tissue resident memory CD8 T cells or TRM cells, talk about the cytokine effects on tissue epithelial and stromal populations, and then close on immune and resonant cell crosstalk in entheses and bone. This slide illustrates the tissue complexity that we encounter in psoriatic disease. I'd like to make 3 points from this slide. Number 1 is that a number of tissues are involved in psoriatic arthritis, ranging from the skin, the bone marrow, the gut, the entheses, and the joints. Number 2, there are a number of parallel pathways that are operative in these different tissues, which include, of course, TNF pathways as well as the IL-23, IL-17 pathway. And there are various phenotypes that one can see in the different tissues. I suspect that there are also different pathways of activation in the different tissues, even though there's overlapping of cytokines. For example, in the skin, we have a dominant phenotype of keratinocyte biology, whereas in the joint, we're talking more about resident fibroblasts, macrophages, and osteoclasts, as well as cartilage and chondrocytes. So there are contrasts, and not only the immune cells and the resident cells, but also likely in the types of cytokine pathways that are being activated. So so-called endotypes, which are driving phenotypes. There's also great plasticity within the cellular compartment. And here we see a Th17 cell. And unlike Th1 and Th2 cells, which have very specific transcriptional programs that are highly regulated, for example, a Th1 cell with gamma interferon, the major cytokine, and Tbet. In the case of Th17, there are a variety of different transcriptional factors that can be activated that can lead to the release of a whole array of cytokines, which can have signals that drive pathogenic events, inhibitory events for pathogenesis, and then non-pathogenic signals. And you can see here the different transcription factors. One can see here the variety of cytokines that can be released. And so just because a cell expresses CCR6 and other markers of being Th17, that does not necessarily tell you what this cell is doing. In order to understand that, one needs to look at single-cell transcriptomics. So this is a really important point, and that's why the complexity both at the tissue level and the cellular level is really quite high and requires special techniques to understand what's going on in individual tissues in individual patients. So here's an example of how monocyte macrophages can take on different roles within the synovial tissue. In a beautiful work published by Kuhlmann and others, they show that in the synovial lining, there are macrophages shown here in red, and these macrophages are forming a barrier, a protective barrier for the synovial lining cells of fibroblast-like synoviocytes in blue shown here. And you can see in animal models of arthritis that this barrier is disrupted, and now these cells are moving out into various inflammatory tissues or pannus. Here is a very beautiful electron micrograph showing these monocytes, which actually have barrier-like features you might see in epithelial cells, and adjacent to them are fibroblasts and connective tissue in the subsynovial area and also in the other part of the lining. It's well known from work that they showed that these cells derive from CX3CR1 monocytes and that they can replenish those monocytes that are present as barrier cells in the lining coming from the subsynovial tissue. Well, CX3CR1 cells are not only in the barrier in the joint, but in this study they showed that this population is a source of tissue-specific osteoclasts. So focusing on the synovial lining in this mouse model and performing single-cell RNA-seq, they were able to isolate this P1 population that had features of osteoclasts. And in additional work I don't have time to go into, they were able to show that this osteoclast, like those border monocytes, derive from CX3CR1 monocytes which come to the joint, and depending on the signals that they receive, specific chemokine signals, or in the case of the osteoclast precursor, MCSF followed by rank ligand, they can develop into tissue-specific osteoclasts that are specific to the arthritis tissue and are different from those osteoclasts we see in other sites of the body. So here again is this complexity and plasticity which comes up at the tissue level as well in the cellular differentiation pathways. So let's talk briefly about tissue-resident memory cells. These are cells that obviously are T cells, and they migrate to a tissue and they stay in that tissue. They're resident, they do not leave. They express CD69 and a subset of them express CD103. They provide rapid on-site response and reactivation. Their highest number at sites of primary activation, so sites where you might have had some type of cut or inoculation or insult in the lung or in the GI tract. And also the joint, as you'll soon see. They're long-lived, but they do not recirculate, and they're antigen-specific. They have alpha-beta receptors that are specific in the skin, the gut, and the lung for a variety of pathogens as shown here. These are the cell surface markers you can see. I'm focusing on the CD8, IL-17 positive cells, which as you see will be important in psoriasis and PSA. They express CD69, as I mentioned, which is important for inhibiting them from leaving their tissue. It blocks the CYP1 receptor. The T cell receptor, which is an alpha-beta receptor. In this case, the cells we're going to be looking at are CD8. They also express CD103 and interestingly PD1, which in other T cells can be a marker of exhaustion along with granzyme B, which these cells can express. But these markers are not ones of exhaustion in this cell subpopulation, but ones of activation. These cells are extremely activated and can be pathogenic. And this slide illustrates that fact so that they're known to have important roles in protecting against pathogens, long-lived responses, whereas they can also participate in allergy, injury, and autoantigen exposure, and they have been shown to be critically important effectors in psoriasis, and I'm about to show you psoriatic arthritis and many other diseases. So this study showed that IL-17 positive CD8 cells seem to be preferentially found in the synovial fluid rather than the peripheral blood of patients with PSA. They found similarly that these cells seem to congregate in the synovial fluid of patients with other forms of spondyloarthritis. Notice that in rheumatoid arthritis that is not the case. Now what's really intriguing is that these CD8-17 cells, a significant subpopulation are CD8 positive and are expressing the T-resident memory cell phenotype, both in the CD8 and the CD4 population. And particularly the CD8 cells are polyfunctional in their ability to produce a variety of cytokines, including interferon gamma, TNF, GM-CSF, and IL-21, in addition to expressing IL-17. So these cells have the potential to be major effectors in the skin and or the joint. So this was a very important study by Penkava in which they removed cells from the synovial fluid and the peripheral blood of patients with PSA, and they performed CyTOF, mass cytometry, to look at the different populations. And here you see memory CD4, memory CD8, naive CD8 cells, as well as monocyte macrophages, NK cells, and B cells from both the peripheral blood and the synovial fluid. When they broke this down into synovial fluid versus peripheral blood, they found there was a very marked expansion of the CD8 cells, particularly memory CD8 cells, to a lesser degree memory CD4 in the synovial fluid. They then went on to perform single-cell RNA-seq on these synovial fluid cells, and once again you see a number of cells that are expressing high CD8, transitional levels of CD8, and then this population here, which is a CD8 T-resident memory cell population, and indeed this was the population that was most expanded in the synovial fluid. You see T regulatory cells here, not so strong. There's some mate cells, and gamma delta T cells not really a major contribution to the synovial fluid in PSA. They then did T cell receptor sequencing of these CD4 and CD8 T cells, and here you see where they found most of the clones, which were in the CD8 and the T-resident memory cell population. And then over here, they looked at those clones that were significantly expressed in the peripheral blood or in the synovial fluid. The red are those cells that are CD8. The triangles, the circles are the CD8. Triangles are CD4. So you can see most of these cells that are clonally expanded are in a CD8, and there's a high percentage of them in the synovial fluid, lesser so in the peripheral blood. So what about cytokine effects on tissue epithelial and stromal cells? Well, I'm not going to walk you through this whole paradigm of psoriasis. I'm sure you know it well, but the key effector cells are obviously T17 cells, which release IL-17 and IL-22, Th1 cells, gamma interferon, IL-12, and then you have this very important subset of T22, and these cells release IL-22. And it's well known in psoriasis as IL-22 is a major driver of keratinocyte proliferation. And these cells, keratinocytes, release a variety of cytokines and growth factors that stimulate the feed-forward inflammation that we see in psoriasis. So in psoriasis, IL-22 is a key cytokine that has very prominent effects. Interestingly, though, when you look at IL-22 in the synovium, it's elevated in the synovial tissues, and yet its effects are not nearly what we see in psoriasis. Indeed, if you knock out IL-22 in many mouse models of arthritis, you see no change in the phenotype. While it does have effects on cells in vitro, it can promote bone formation or bone resorption in assays, in vitro assays, and proliferation. And when in synergy with TNF, it can drive very high release of a variety of cytokines, IL-1, IL-8, etc. Yet in the synovial tissue itself, it does not appear to have a major effect, in direct contrast to what we see in the skin. So once again, tissue-specific events with the same cytokine. The other important area to mention are the stromal cells, which heretofore have not received enough attention. And this very highly quoted paper, beautifully done, Croft and others showed that there were distinct subsets of fibroblasts, and these were located in the subsynovium, and they were expressing Thi-1, and they drove inflammation, whereas the Thi-1 negative fibroblasts, which were located in the synovial lining, were releasing metalloproteinases and expressing rank ligand and driving joint damage. So here you have the important point that topography is essential to understand in order to gain insights into how the cells are interrelating with each other and carrying out various effector functions. Well, the other important point regarding spondyloarthropathy in stromal cells was published in a paper by Dominic Baton many years ago. And basically what he did was to take synovial tissue from patients with SPA in blue on the top there and patients with RA in orange-yellow and perform transcriptomic analysis focusing on muscle and myofibroblast genes. And what he showed clearly here was that the SPA tissues strongly express myofibroblast muscle genes in direct contrast to RA synovial tissue. So I think this is an area we should focus on more directly. We certainly have a variety of agents that target immune cells and immune cell cytokines, but this is an area that we may be able to gain even more handle on and improve our responses in spondyloarthritis. What about immune and resonant cell crosstalk and entheses in bone? Well, this is a functional model of enthesitis which really incorporates all of the features. The mechano sensation and immune activation with focusing on innate immune cells, likely acquired immunity is important here too, with the type 17 cells, Th17 and type 17, the innate immune inflammatory response, and then you have the mesenchymal proliferation, in part driven by the release of PGE2 and IL-23 and other cytokines, which then ultimately converge to result in bone remodeling through the activation of these cytokines and growth factors on monocytes to drive osteoclastogenesis and on osteoblast precursors to promote pathogenic bone formation. I'd like to talk now about mediators of osteoclastogenesis, which you know well, TNF, rank ligand, and IL-17, but it turns out these particular cytokines can have Janus-like properties. In other words, not only do they necessarily drive the resorption of bone, but they can actually also drive, in certain circumstances that we're just learning about, new bone formation. So here you have a paper from China where they looked at Wnt proteins in the spinal ligaments and sera of AS patients. And they found in this paper that a variety of Wnt proteins were elevated in both the tissues and the sera. They then went on to address the question as to whether low concentrations of tumor necrosis factor may be as important in driving osteoblast differentiation. And here you see that these are unstimulated, these are mesenchymal cells shown here, or osteoblast precursors, and they gave conditioned media and then increasing concentrations of TNF. And they were able to show, as reflected in the bar graph below, that the osteoblast formation was increased in doses of TNF between 1 and 10 nanograms per mil, but not at the higher dose. And so they propose and show the data in additional experiments in this paper that low concentrations of TNF can actually drive the differentiation of osteoblast progenitors. And over here, they look at Western blot analysis to show that in these are raw cells, which are an osteoclast precursor line, and monocytes, that when you treat them with no treatment versus TNF, again, these are low concentrations, that you can see an upregulation of the various Wnt molecules that they found in the sera and the spinal ligaments of these patients. So they hypothesized that low levels of TNF that you might see after you're treating patients with anti-TNF agents may be essential in driving continued new bone formation. In another paper, these authors showed that rank, which is released in vesicles, and the rank here is shown as sort of an antibody, that rank released in osteoclastic vesicles comes out and then can actually bind to rank ligand, which is on the surface of osteoblast precursors, and through reverse signaling in the mTOR pathway, upregulate RONX1 and drive bone formation. So once again, I'm coming back to that theme of complexity, plasticity, and the tissue microenvironment, which is driving pathologies which include synovitis, bone loss, new bone formation, enthesitis, and we have to understand what's going on at the tissue level with the specific cells at transcriptional understanding of how these events are unfolding in individual patients. So in conclusion, I've emphasized plasticity, which is so important at both the cellular and tissue level. I've discussed the important role of T-resident memory cells, which are known to be very important effectors in psoriasis. I didn't have time to talk about that, but that's been published by Rachel Clark and others, and in PSA. And there's also support now in RA and juvenile arthritis. The differential effect of cytokines on tissue epithelial and stromal populations with synergies and tissue-specific actions. And lastly, immune and resonant cell crosstalk, talking about the importance of biomechanical stress, TNF, rank ligand, and IL-17, which may have opposing properties in terms of their effect on bone, depending on the individual tissues and the microenvironment.
Okay, so we're going to do the post questions. I'm sorry there have been some technical issues we're dealing with. And so, first of all, which statements about tissue-resident memory T cells is correct? First, A, long-lived cells that continuously circulate through non-lymphoid tissues. B, have a limited T cell receptor repertoire. C, are lymphoid cells but do not have a T cell receptor. Express CD69 and CD103. Provide a very rapid antigen, non-specific cytokine response to local injury or infection. Jörg, I'll let you do the countdown.
5, 4, 3, 2, 1. Okay. Do you want to comment on the result?
Yeah, absolutely. So the majority got the right answer, and that is that they express CD69, which keeps these cells in the tissue, and 103. They express alpha beta receptors. They're rapid reactors to specific antigens, and as discussed in the presentation, it looks like they're major effectors in psoriasis, PSA, RA, and juvenile inflammatory arthritis. So really important targets as we move forward to improve outcomes in these diseases.
Yeah, having you, on camera here, let me just ask you an immediate knee-jerk question. So are CD69 and CD103 potential targets for monoclonal antibodies?
Absolutely, yes. And people are looking at this now, especially, in animal models. One of the big questions is what drives the production of these T-resident memory cells in human tissues? We don't know. In the mouse, it looks like it's IL-15. And the other question that comes out is when these cells first circulate and then get to the tissues, what is the differentiation program that allows them to express CD69 and 103 and stay in the tissue and not migrate? So these are really critical questions that we're looking to find answers for. And obviously therapeutic approaches that are looking at 69 and 103 are really attractive targets.
Okay, thanks. Next slide. Which statement describes best the role of IL-22 in psoriatic arthritis? A, it provides key proliferative signals to dermal fibroblasts in the skin. B, it's a major mediator of pathologic new bone formation in the spine. C, it's a critical activator of osteoclasts in peripheral joints. D, it mediates the recruitment of Treg cells to sites of inflammation. Or E, may play a limited role outside of the skin. And you have 5 seconds. 5, 4, 3, 2, 1.
Yes, again, majority got the right answer. I think, Luis gave a beautiful talk on psoriasis and mentioned the important contribution of IL-22 to keratinocyte proliferation. I put this question in here because if you look at the in vitro literature, there's a variety of things that IL-22 does to bone cells and to fibroblasts, et cetera, that can be quite impressive, especially when you add a little TNF there. But if you look in the models, and this is work from Helen Benham and also Lubert's lab, it doesn't seem to play a critical role in driving these cells in vivo. So it just illustrates one of the points I was trying to make in the talk is that it depends on the tissue environment as to what that cytokine is doing and what are the resident cells that it's acting on in order to understand what the mechanisms, the pathophysiology might be.
Yeah, and I want to add that this is also an intentional trick question to some degree, because I think that many got hooked on the key proliferative signal, which is true, but not for dermal fibroblasts.
Right.
The target cell in the skin are the keratinocytes and not the fibroblasts.
Correct.
Okay, so I think then we will go to our Q&A and we'll hopefully welcome the other panelists. And while this is happening, I would actually like to say a special thank you to Kurt and Louis in Europe, because at your location, it's in the middle of the night right now. So thank you for staying up.
Almost.
So I want to start this panel discussion with a question to all of you. What do you consider sort of the major gaps right now in understanding the pathogenesis of psoriasis, psoriatic arthritis, or axial spondyloarthritis?
I think that the complexity and heterogeneity are key barriers to our understanding. I also think that there may be more, and this is my own opinion based on recent literature, that the acquired immunity might be much more important in these diseases than we thought. And that we have not identified, certainly we've identified autoantigens. LL37, as Luis mentioned, has also been identified as an autoantigen PSA, psoriasis and lupus. The ADAM TS5 molecule as well. And then, as we know, and Kurt was mentioning this when we were chatting behind the scenes here, that there are peptides that have been recently published in a paper in A&R that may serve as autoantigens. So I think trying to understand the key pathways that might be driving this, which have some acquired elements to that, and also trying to understand the complexity and heterogeneity with our newer techniques, single-cell RNA-seq, eSite-seq, I think are going to provide a lot of important insights into these diseases.
And in psoriasis, there are perhaps many unresolved issues, but we need to understand better the missing link between local skin inflammation and systemic inflammation. That's very important. And on the other hand, it is also quite important to find a way to prevent tissue-resident memory cells to become installed and to reactivate psoriasis in time. And we have seen, we've been puzzled by the long remission which we can achieve when we block interleukin-23.
I think that one of the main remaining questions is what kind of inflammatory pathway is driving psoriatic arthritis and spondyloarthropathy, because in contrast to what we see in rheumatoid, for example, CRP levels are quite low, so meaning that at least that pathway is not the dominant inflammatory pathway driving the diseases. And I think it's still a lot to work about identifying the clear inflammatory mediators in this disease group.
Obviously, the other really important question is how can we in rheumatology match those marvelous outcomes that Louis showed in psoriasis? We're far from that. And if you look at the first trial of anti-TNF agents, which was etanercept by Philip Meese in 2000, the outcomes we're seeing now in PSA are of the same magnitude. Whereas in psoriasis, we've seen, as Luis beautifully illustrated, dramatic increases in response. And why is that, and how can we achieve those better responses in a joint?
So maybe this is partly explained by the type of instruments we're using to evaluate disease treatment effect. Of course, let's say PASI evaluation, at least to me, seems a quite objective or more objectable outcome, as we are using these composites where patient global and pain plays an important role in the instrument. And we all know that it's only recently that we try to characterize the type of pain which is present in this disease.
Too high placebo response on one hand. On the other hand, most of the trials are done in peripheral polyarticular patients, which are a real minority. This is not the way in which the disease starts in our patients.
Yeah, I want to add to this. I think I was also a little jealous about the elaborate mechanistic cartoons that you showed about what's happening in the skin, because we don't have anything like that for what's happening in the spine, for instance, or in the SI joints in spondyloarthritis. And that's also something to do with our ability to actually look at what's happening in the tissue. And the skin is easily biopsied, and we have very little information of that kind from the SI joints or the spine. Neti, may I ask you, so how do you communicate genetics to patients, you as a patient and physician, and the complexities of what we know about the interplay of genetics and environmental factors? Because people often ask that, and there's not a lot of time in the clinic.
There's not, and I think that it ends up being quite high level in terms of the potential for genetics to have a role. Clearly, genetics becomes more of an issue if they already have family members or there's a concern or the questions come up related to risk. I think that one gap that exists, for example, if we look at the trials of major therapeutics and the approvals on which they were based, there is underrepresentation in terms of diverse individuals. So we also don't always know if the therapies will work for all of the patients that we're seeing, because as we know, clinical trial populations reflect a very small percentage of the patients that we might see in the clinic. So all of those things come up into the discussion, and then weighing the various options, related to if you have a psoriatic arthritis patient with dactylitis or enthesitis or spinal disease, and trying to balance all of those things with the comorbidities. And you inevitably see, as you know, patients where the skin's getting better, but the joints are not, or vice versa. And then you're having to figure out what are the best therapies for them in those cases.
That's a perfect segue to my next question, and maybe that goes to Kurt and Chris. So IL-23 inhibitors don't work, or at least 2 clinical trials in AS and axial spondyloarthritis failed, even though ankylosing spondylitis is very strongly associated with multiple genes in the IL-23 receptor pathway. Which explanation for that do you favor at this point?
Kurt?
Well, I think one of the ideas is that IL-23 might play a role in the beginning of the disease and in the initiation of the disease, but not in the maintenance of the disease. And of course, we all know that the delay in between first symptoms and diagnosis is still quite high. For spondyloarthropathy is still around 4, 5 to 6 years, and even in psoriatic arthritis, even in our region, it's still like we have a mean delay of like 1 to 1.5 years. So I think that might play a role in the differentiation. I think secondly, I think from a pathogenic insight, it's still not very clear is how many of the diseases enthesial and how many of the diseases synovial driven. So, and that might also be one of the key differentiators why they are not so responsive to IL-23. And again, to come back to what Niti mentioned, is that I think there is still a problem with the clinical phenotyping, even within the same concept, that not all these patients have at least the same pathogenic or the same mechanism for developing, let's say, a kind of final stage of symptoms. After all, I always mention that you can have many causes or many processes in a knee, but the end is always knee pain and knee swelling. And behind there might be many different processes. And I think as the delay in these diseases between symptom and diagnosis is so long, that might play a role also.
Yeah, so I agree with everything Kurt said. I think the paper by Yeramenko, which he quoted, which is it's important early in the disease and maybe not later. The other explanation are there are innate immune cells that produce IL-17 independent of IL-23, and they could be playing a larger role in axial disease, possibly. There's two questions, one from Judy and one from Maureen, you might want to address.
So there's a question about the role of IL-36 blockade in psoriatic arthritis. And generally, what is the state of, because that's for the rheumatologists, that is not a well-known target. So what is the state of IL-36 inhibition?
I'm going to turn to Luis for that.
Yeah.
Well, no, it's whenever there are polymorphonuclears, IL-36 is important because it's the main driver of the activation of the keratinocyte. But it seems to play a role also in perhaps synoviocytes and other cell types in rheumatology. So really, keratinocyte is a huge producer. But the interface which really matters in rheumatology might be the synovium. I think it makes more sense. There are much more polymorphonuclears in a pannus or in the inflammation in the synovium than on the enthesis. That's my belief.
Judy has a question about CD69 as a general T-cell activator marker. That's true. There are other more specific T-resident memory markers. ZF, I think it's 486, which was actually in the paper that I talked about by Podkova, is one. And there are other markers that are more specific for these T-resident memory cells we're now learning about. She's absolutely right that there's better markers now.
I think we have to come to a conclusion of the session. I will ask one more question. There's currently in the field a big debate about the relationship between axial psoriatic arthritis and axial spondyloarthritis. From the pathological or pathophysiological perspective or genetic perspective, can you comment on that debate?
Well, I think the debate, this comes down again to outcome measures. So the debate has really been generated by the Discovery 1 and 2 trial, where they showed that when they use, quote, axial measures in a peripheral arthritis population, that they seem to see better responses in the patients that received guselkumab either at 4 or 8 weeks than the placebo. And we've had huge discussions about this. I'm sure everyone else around here has as well, that these measures are really not really specific for axial disease, number one. And number two, the patients that were selected for this study was based on films that were not centrally read. And we all know that in axial SPA, that is a problem. So I think the answer remains to be determined. I think that you're going to need a trial like they did with secukinumab, where they actually did very imaging that was centrally read and carefully following these patients to answer that question. And I think that'll let us know whether or not there are differences between axial PSA and axial SPA, which I personally think is unlikely, but we'll see.
Might it be that let's say the cells at work in the bone cartilage interface are different from those in the bone tendon interface?
Yeah, it's very possible. I don't disagree.
And also in the spine there also there's synovium, there are the facetal joints, so there might be a bit of a composite of both mechanisms.
Mm-hmm. Possible. Absolutely possible.
Yeah, but the facet joint is a quite strange joint because one of the sides is purely ligament and the other side is synovium. So, but I think one of the problems is that axial disease has not properly been studied in psoriatic arthritis. There are some old, long-dated radiographic studies, and that's it. And if you look in populations and you ask for axial symptoms, a lot of patients will report axial symptoms. But it doesn't still mean that they have axial disease, because the inflammatory back pain resides among all kinds other back pains which are much more prevalent than inflammation. And I don't know why those patients wouldn't have not the right to have, let's say, the classical type of back pain also. So. And it's true, like BAS-DAI is a very bad measure to differentiate between inflammatory back pain and other types of back pain. So I think there are a lot of, let's say, conceptual issues of all the data reported up to now. So to sort this out, more appropriate studies should be done.
So it's still a lot of things that we need to learn. With that, I would like to thank you, you panelists, for participating in this seminar and for all your work in preparing it. I would like to thank Lisa with Spartan and Janine from Grappa for their hard work in getting this organized. I would also like, again, to thank Pfizer for financial support and Nucleus for making this virtual event possible. I also would like to thank all the people in the audience whom we don't see for participating and for staying up if it's nighttime where you are. Lastly, and maybe can go to the next slide. I would like to alert you to another 4 upcoming seminars in March, April, May, and June. You can sign up for this through the website. If you found the first and the second seminar informative, then please spread the word. The recording of today's seminar will also be available through the Spartan and Grappa websites. And my apology for a few technical glitches that we had along the way today. Lastly, please let us know in the evaluation what you liked about the seminar or what we can potentially do better in the future. Thank you very much, and have a good day.
Thank you.